Hello and welcome to Sam Biology class. Friends, today we shall discuss about nitrogenase complex. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Biological nitrogen fixation is catalyzed by metal enzyme known as the nitrogenase complex. It is extremely oxygen sensitive and catalyzes the ATP dependent reduction of dinitrogen into ammonia. Three distinct types of nitrogenases have been found in diazotrophs. They are number 1, the molybdenum nitrogenase, number 2, the vanadium nitrogenase, and number 3, the iron nitrogenase. All three are two-component systems. They are structurally and mechanistically related but genetically distinct as they are encoded by separate gene clusters. First one is the molybdenum nitrogenase, which is the most widespread and intensively characterized nitrogen fixing enzyme. It is encoded by the gene cluster NIF. Various structural components of this enzyme are encoded by genes such as NIFH, NIFD, and NIFK. The molybdenum nitrogenase consists of two components. One is the Fp protein and the other component is the MOF protein. Fp protein is the smaller dimeric component. Its molecular weight is about 60 kilodalton. It is a dimer of two identical subunits which are encoded by the gene NIFH. The Fp protein symmetrically coordinates one iron sulfur cluster with the ATP binding sites at the dimer interface. The other component is the MOF protein, which is a tetramer consisting of 2 alpha and 2 beta subunits. The total molecular weight of the MOF protein is about 230 kilodalton. The crystal structure of MOF protein is alpha 2, beta 2, heterotetramer. The alpha subunit is encoded by the gene NIFD and beta subunit by NIFK. The MOF protein contains two complex iron sulfur clusters in each alpha beta dimer. The MOF protein, which is the catalytic component, contains two complex iron sulfur clusters in each alpha beta dimer. One is the P cluster that remain buried at the interface between alpha and beta subunits. It serves as the initial acceptor of electrons from Fe protein. The other cluster is the MOF cofactor that remain buried within the alpha subunit. It represents the site of substrate reduction. The function of MOF protein is to reduce dinitrogen into ammonia. Hence, it is also called dinitrogenase. On the other hand, the function of the Fe protein is to catalyze ATP-dependent electron transfer to the MOF protein. Hence, it is also called dinitrogenase reductase. In the overall nitrogen reduction reaction, ferredoxin serves as electron donor to the Fe protein, which in turn hydrolyzes ATP and reduces MOF protein, which finally reduces dinitrogen into ammonia. Binding of ATP to the Fe protein is thought to cause some conformational change which facilitated the transfer of electrons. The other two types of nitrogenases are the vanadium nitrogenase and the iron only nitrogenase. The remaining two nitrogenases are the vanadium nitrogenase and the iron only nitrogenase. Both are two component systems. The vanadium nitrogenase is encoded by the gene cluster VNF, and its structural components are encoded by specific genes such as VNFH, VNFD, VNFG and VNFK. Third one is the iron only nitrogenase, which is encoded by the gene cluster NF, and its structural components are encoded by genes NFH, NFD, NFG and NFK. So friends, this is all about nitrogenase. For more updates, subscribe my YouTube channel, Sam Biology Class.